In this video, we will discuss about nucleic acid and lipid nanoparticles related therapeutic approach. So we will first discuss about this nucleic acid related therapeutic approach, or we can say that RNA related therapeutic approach. So it can be used for the production of therapeutic proteins which cannot be uh, produced in the patients, and it can supply the sufficient amount of therapeutic proteins in case the productions of that protein is low in the patients, that is causing them some kind of problems. And if the gene is mutated in the patient, then they may not be able to produce the correct type of protein. In that case, we can use the RNA or correct RNA sequence of that protein and produce the proteins through the produce these proteins or the wild type protein, and then can supplement those protein requirements in the patient. And if suppose in the laboratory it is not or ex vivo it is not possible to make stable proteins, then we can use this RNA sequence directly into the patients to form this kind of proteins that are unstable in the ex vivo conditions. If some new kind of proteins or exogenous version of the proteins that is not usually found in the patient, in order to diagnose themselves can also be produced through the RNA sequence, and RNA can be used along with this CRISPR-Cas9 technologies to target a specific part or specific tissues or organelles of the patient, and it can also prevent the formation of this mutant protein synthesis, either by interfering with their splicing process, or by using the CRISPR-Cas9 to edit or remove these genes. And the interference with endogenous nucleic acid exon skipping. So this is about exon skipping or DNA editing. Step 8 or the strategies 8, or in the strategy 7 is about this prevention of mutant protein synthesis. This can be done by one of the approach that we have discussed yesterday, and just for the revisions point of view, this mutant protein synthesis can be prevented by using uh, the mRNA that can attract RNAs, so that this mRNAs of the mutant proteins will be degraded. Now we will also discuss about some of the lipid nanoparticles delivery vehicles. This has recently been uh, gained much attention, and some of the FDA approaches are also been approved, such as the partisans, where this silencing RNase is used to treat this amyloidosis, and this elasorman, in which case one of the example is this mRNA related COVID-19 vaccines, and this dosirman also is one of the examples of this mRNA related COVID-19 vaccines. And in this 2002 NTLA, the Cas9 based mRNA and sgRNA gene editing therapies for hereditary and angioedema diseases. So this has also been approved by the FDA. So what these lipid nanoparticles consist of? This consists of this ionizable lipid, cholesterol, helper lipid, and this PEG lipid. And combinations of this, or we can say that mixing of this, will help in the formation of lipid nanoparticles that can deliver the target gene to the site. So what happens is that rapid mixings of these four components are required, and in that case what will have the one things that when we have to understood is that as the mixing continues, the organic solvent polarity increases, and that helps in the formation of this hydrophobic PEG lipid or we can say that the hydrophobic PEG lipid will form a final layer around the lipid nanoparticles that can be seen here. It usually forms a size of 100 nanomolar, and in that case the lipids which are usually present in the organic solvent will, when it moves into the acidic aqueous solutions, then it can be easily ionized or protonated, and that leads to this formation of these circular structures. And the PEG lipid, which is also hydrophobic in nature, once it is enter into the or mix into this ionizable or aqueous acidic solutions, then it will also form a final coating layer around these lipid nanoparticles. So these lipid nanoparticles in finally can be used for the treatment of diseases. That some of the examples are shown here in the first row of the figure in the right hand side. So these are some of the therapeutic approaches based on nucleic acid or RNA and the lipid nanoparticles based. If you need to understand more about this therapeutic approaches in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.